Good evening, students. In our last class, we learned about the type of reproduction, that is binary fission in bacteria. How bacteria divide and reproduce is given. Here you can see first one bacterial cell. You can see a circular DNA inside the bacterial cell. That first it to duplicate or replicate, make its own mirror image like structure, it make duplicate, then that will move apart. That copy of the DNA may move apart, then a constriction appear in between these two DNA structure, and that will grow more deep, and two cells are formed. That are the type of reproduction seen in bacteria, that is binary fission. This kind of reproduction is known as binary fission in bacteria. Then again you can see how it reproduce, how much bacteria are produced every day by it will take 20 minutes or it is considered 20 minutes or half an hour for to complete a bacterial cell to complete its growth and to reproduce. That will take only half an hour for a bacterium. Then how much bacteriums are produced are given a very big number that is given here. It's difficult even to read that. More than crores of bacteria may be produced. Thousands crores of bacteria are produced within 24 hours. But in the environment, it is controlled by the sunlight. That is why the bacterium cannot multiply and that cannot harm our environment, because certain unfavorable conditions, if this bacterium exposed to the sunlight, that may get destroyed. That is how the bacterial cells may reduced in the environment. By if all the conditions are favorable, bacterial division within a single bacterium may divide, and you can read the how much it may be produced in the environment within a single bacterium can multiply into this large number you can read. Then another type of reproduction, that is sexual reproduction, also seen in bacteria. Here that type of reproduction is known as conjugation. You can see the word conjugation. Then that is two different strains of bacteria. Strain meaning positive, negative, male or female kind. There is no male and female really. That is known as different strains, positive or negative kind of bacteria. Considered it is male or female. They come together, and there may be a tube formed in between, and the plasmid meaning the round DNA is known as the plasmid of the donor bacteria. The one strain positive may be transferred to the recipient through a hollow tube like extension of the donor. I shall show you the picture of the conjugation by writing. After this class I shall put a picture how the conjugation occur. Two bacterium cell come together parallel to each other, and a tube form in between, and its round DNA may be transformed from one bacterium, one strain to another. In the same species, it occur in the same thousands of varieties of bacteria are present. In the same species, bacteria may do this kind of primitive type of reproduction. The conjugation is a primitive type of reproduction. Then another important thing happening to the bacterium in the unfavorable conditions are spore formation. Spore to overcome unfavorable condition. You can see the bacterial spore here. really the bacterial wall you can see in the picture. You can see the bacterial wall in the unfavorable condition when at the time of drought, or there is not enough water, or in the frozen conditions. In such cases, the bacterial content will shrink and make a spore, and spore build a wall around it. Bacterial spore wall may be formed around it, so that it will survive. Here certain facts about that is given here as the point. You see, these can tolerate extreme dryness, and some cannot be killed even at the temperature of boiling water or frozen ice. This spore can, when we drink the boiled, sometimes we may drink boiled water. We may boil water to kill the bacterium present in it, but you must understand that the bacterial spore can survive even the temperature of boiling water or frozen ice. That spore can remain. If it reach our stomach, or when it reaches the favorable condition, it can again come out by breaking its spore wall and can act like a bacteria. Then some bacteria can tolerate poisonous chemicals, so that in this spore condition it can survive. Many of the bacterium can survive even in the unfavorable condition. That may remain as dormant. Dormant meaning in a sleeping condition it will remain, its protoplasm remain there in a dormant condition. When it get favorable condition, this will become active. Then again, then another thing you have to notice here is that spore formation in bacteria is not a method of reproduction, but simply a method of escaping from unfavorable condition. We cannot say that the spore has a mode of reproduction, but the same bacterium, reproduction meaning production of another individual from a living being, that is the reproduction. But the spore formation, so that we cannot tell it as a mode of reproduction. It is only the same bacterium is remaining alive without dying, and it overcoming the unfavorable condition that is happening in the spore formation. So we cannot consider it as a mode of reproduction. Then again, you can see the useful role of bacteria in medicine. You know that many in the medical field, bacteriums are very useful. Bacteria are used in medicine in the production of antibiotic, serum and vaccine. Antibiotic meaning, antibiotic definition is given here. You can read it. An antibiotic is a chemical substance produced by a living organism. Biotic meaning living enna. So antibiotic is that is produced by that is a chemical substance. Antibiotic is a chemical substance that is produced by a living organism. That is not artificially made from any other thing. It is made out only from a living organism that can stop the growth of some disease producing bacteria or fungi. It can destroy the growth of another bacteria or another fungi. Antibiotic may be produced from bacteria as well as in fungus, but it can kill another bacterium or another fungus. It can used as a medicine to kill another harmful bacteria. That is antibiotic. Then certain important, numerous, many antibiotics are there, but important antibiotics are given. Certain names of important antibiotics are given here. You see streptomycin. You might have, when you buy medicines from the shop, antibiotics, you might have observed this name in the medicines. When you bought it from medical shop, streptomycin, chloromycetin, tetracycline, penicillin, etc. are the names of certain important antibiotics. Then here penicillin, here the first produced antibiotic is the penicillin. Here penicillin really made from the fungus, not from bacteria. Fungus, mold, from a mold penicillin is produced. Its discovery, the story of that discovery of penicillin is given. It is an interesting story, so that we can read it. In 1929, Alexander Fleming, a scientist, observed that some bacteria growing in the culture were destroyed by an accidental contamination of mold. Culture meaning for different experiments, usually scientists are growing this microorganism. Culture meaning they are cultivating this microorganism in small dishes or in a flask. They are keeping it in the lab. In such a culture were destroyed by an accidental contamination of mold penicillium. There were many bacterial culture or virus or mold cultures were there in the laboratory. Then it is heard that when this penicillin, that culture of the Penicillium notatum was happened to spill to that some bacterial growth. അത് അറിയാതെ കയ്യിൽ നിന്ന് പെൻസിലിൻ നൊട്ടേറ്റം അത് ആ ഫംഗസ് ബാക്ടീരിയ വെച്ചിരുന്ന കൾച്ചറിനകത്തേക്ക് വീഴാനിടയായി എന്നാണ് പറയപ്പെടുന്നത്. ഹാപ്പൻ ടു സ്പിൽഡ് ടു ദ, ദാറ്റ് ഈസ് കണ്ടാമിനേഷൻ, accidental contamination of a mold Penicillium notatum. Then Alexander, meaning here Alexander Fleming discovered that this mold produced a substance which suppressed the growth of many bacteria, and the substance was named penicillin. That substance that is produced by the Penicillium notatum. Penicillium notatum is the scientific name of that mold. That culture, when it happened to spill into the bacterial growth, many bacteriums were killed by that substance. So he named that substance that produced by the Penicillium notatum as penicillin. Penicillin എന്നാണ് ആ ആൻറ്റിബയോട്ടിക്കിന് ആ ഒരു substance that is produced by the mold. Then the first antibiotic for medical use, but remember penicillin is the product of mold and not of a bacterium. Then the next antibiotic, the second invented antibiotic was the streptomycin. It was discovered by Selman Waksman in a similar way. Its source organism was bacterium Streptomyces griseus. You see here the second antibiotic streptomycin was produced by the bacterium, but penicillin is produced by the Penicillium notatum fungus. That is how it happened to invent. Then here some well-known antibiotics and their sources and the disease treated by them is given here. It is for your knowledge, it is given here. Then penicillin, we read about it. Penicillium chrysogenum was the mold, its scientific name. Then it is the disease treated was tetanus and diphtheria. Diphtheria is a kind of fever with cough. Such symptoms are there in the diphtheria. Tetanus meaning you know that when we happen to fall in the soil, the iron bacteria in the soil, or we know that the iron nails, if it happen to make an injury in our, or any iron substances happen to make injury on our hand or on our body, this bacterium may cause the kind of fever known as tetanus. That is a deadly disease. We know that we usually take TT injection to prevent this tetanus bacterial growth. So the organism, the injection is for Penicillium chrysogenum. Penicillin injection antibiotic is given for the tetanus and diphtheria. Then streptomycin, its source organism Streptomyces griseus, that is tuberculosis. Tuberculosis എന്ന് വെച്ചാൽ ക്ഷയരോഗമാണ്. Severe cough that can affect the whole body, especially cough and respiratory organs. Its mucous membrane may be affected by the bacteria. So it happens, severe cough, sometimes blood may come out as with the we may happen to vomit and blood may come with it. That is the tuberculosis. The whole body may be affected by the bacteria. Then chlorotetracycline, Streptomyces aureofaciens, that is typhoid. Typhoid meaning that disease with high fever, at the same time diarrhea, together make the disease typhoid. That is the typhoid symptoms. Then erythromycin, that's Streptomyces erythraeus, that is rickettsial fever. Rickettsial, a kind of bacterial fever, that is rickettsial fever. So that many severe diseases can be treated by this antibiotic. Then criteria for good antibiotic. How we can consider an antibiotic or a medicine as a good antibiotic? It should be able to kill a variety of disease producing organism. That is a broad spectrum antibiotic. Several diseases can be treated by a single antibiotic. That is the broad spectrum. Broad വലിയ ഒരു spectrum antibiotic. It can use to kill several microorganisms. That is one of the criteria. Then it should not produce undesirable side effects. That is also a good antibiotic. If it is if we happen to enter the antibiotic as medicine, if we consume it or inject it, in such cases that should not make any undesirable effect in our body. Then it should not kill normal bacteria of the host. We know that in human body we have B complex producing bacteria inside our body. So such bacterium, useful bacteria inside our intestine, should not be killed. Then other uses of antibiotic. We are more than a medicine, we are using antibiotic in several other ways. You see it as food preservative, especially for fresh meat and fish. Even the milk, inside the milk we heard that certain antibiotics are used even inside the milk that when we buy packet milk. Then as food preservative antibiotics are used. Then for treating animal feed, even the animal feed to use as a preservative, otherwise it may destroyed by many fungus or bacteria. Animal feed, for the cattle feed, like cattle feed or poultry feed, that for treating or preserving it, the antibiotics is used. Then for controlling plant pathogens, we know that many diseases may be affect the plant body. So it can also be controlled by the antibiotic. Then kills disease causing harmful microorganism. It can destroy many disease causing harmful microorganism.